Hello, dear students, welcome back. Today, our topic is Wobble Hypothesis. And understanding Wobble Hypothesis is very important to understand the codon-anticodon interaction during protein synthesis. So, let's begin. The Wobble Hypothesis explains basis of codon-anticodon binding. There are 64 codons in our genetic code. Out of these 64, only 61 codons code for amino acid. The other 3 codons are stop codons. They don't code for any amino acid. The third factor is, though there are 61 codons, but we have only 40 to 45 tRNAs in our cell to bind all those 61 codons. That means number of tRNAs is lesser than number of codons. Wobble hypothesis actually explains why number of tRNA is lesser than number of codons. Now, actually, Wobble hypothesis explains the degeneracy of genetic code. Amino acids have multiple codons, but they do not require as many tRNAs. It implies that some tRNA can bind more than one codon. Therefore, fewer tRNA are needed. So, takeaway from this slide is some tRNAs can bind multiple codons. So, Wobble hypothesis was proposed by Sir Francis Crick in 1966. Yes, the same Francis Crick who co-discovered DNA double helix in 1953 along with James Watson. As per Wobble hypothesis, in codon-anticodon binding, the first two bases of the codon pair with anticodon as per Watson and Crick pairing rule. That means A will pair with U and G will pair with C and vice versa. However, the third base of the codon may not follow Watson and Crick pairing rule strictly. It can bind differently. This explains how one tRNA recognizes multiple codons and why there is flexibility at the third base of the codon. The third base can tolerate some mismatch pairing. So this is what the Wobble hypothesis says, that when codon pairs with anticodon, the first two bases will follow strictly Watson and Crick pairing. The third base may not follow Watson and Crick pairing. It can show some different non-Watson and Crick type of pairing or wobble. Wobble literally means play, or you can say mismatch. So in this picture, supposing this is the mRNA where these are all different codons, CCU, CCA, CCC. These numbers represent the position of bases. And supposing this is your tRNA and this is one tRNA having this anticodon, this is another tRNA, this is another tRNA, and this is another tRNA. So these are the bases in the anticodon of tRNA. Let us assign position to this. Since tRNA is read 5' to 3', therefore this is the first position, second position, and third position. 3, 2, 1 respectively. Okay. So we are talking about codon. Always remember to mention codon for Wobble hypothesis. Okay. So the first two positions of the codon in mRNA will follow strict Watson and Crick pairing. But the third position, the base at the third position may not follow Watson and Crick pairing. It can have a non-WC pairing. Okay, or this is also known as Wobble pairing. Fine. So, first and second base of the codon binds anticodon as per Watson and Crick pairing. That means purine will bind pyrimidine, pyrimidine will bind purine. However, the third base may show non-Watson and Crick pairing. So here in this example, the third base is U, and you can see here the first base of the tRNA that pairs with U is I. I means inosine. It's a fifth type of base formed from adenine. Adenine is converted to inosine through an enzyme. Okay, it's a modified adenine, inosine. So, if there is tRNA first position, if there is inosine, it can pair with AUC at the third position of the codon. So, you can see here A is purine, U and C are pyrimidine. Still, they can pair with inosine. That means this is somewhat different from Watson and Crick pairing rule. So let us understand this through this diagram, where this is our mRNA for running from 5' to 3', and this is our codon, and sorry, this should be U. And this is our tRNA having an anticodon IgG. Okay, so as per Wobble hypothesis, codon's third position, that means U, anticodon's first position, that means I. These are the two Wobble positions. And the first and second base pairs of the codon, they will bind anticodon through WSC pairing rule. Whereas the binding between the Wobble positions of codon and anticodon follow the Wobble pairing, as seen in our previous table. Okay, this is what we mean. So let us summarize one more time. Codons run in 5' to 3' direction. Anticodons are read in 3' to 5' direction. That means supposing this is your codon, 5' to 3', CCU, 1, 2, 3. And this is your tRNA, the 5' to 3'. So IgG. Okay. So codon runs in 5' to 3' direction. Anticodon is read in is complementary to the codon, therefore it runs as 5' to 3' in the reverse direction. First two bases of the codon, they are strict pairing as per the Watson and Crick model. The third Wobble position, however, will not follow Watson and Crick pairing. Instead, it will have a Wobble pairing, which can have unusual pairing combinations. Now, Wobble pairing allows non-standard base pairing, and it increases the efficiency of translation. So this table explains the Wobble pairing between the codon and anticodon. The Wobble position, if you remember, is the third base in the codon and first base in the anticodon of the tRNA. So, if we have G in the anticodon of tRNA at first place, it can pair with U or C in the codon third place. If there is U as the first base in the anticodon, it can pair with A or G in the third place of the codon. And if we have I as the first base in the anticodon, then the tRNA can pair with a codon which has U, C, or A in the third place. So this is what is explained by Francis Crick, and he gave this Wobble pairing rules for the third position base in the codon, which will pair with the first base in the anticodon. So let us now take the example of amino acid alanine coded by three different codons GCU, GCC, and GCA respectively. Now, in many of the cells, there is only one tRNA for alanine which can bind all these three codons. So, Wobble hypothesis explains why a single alanine tRNA with anticodon IGC from 5 prime to 3 prime. This is the orientation, okay, from 5 prime to 3 prime. Why a single tRNA having this anticodon is capable of binding all the three codons of alanine. You can see here in this picture because in the first place at the Wobble position in the tRNA. What is the Wobble position in the tRNA? It is the first base. And Wobble position in the codon, it is the third base. Okay. So Wobble position of the tRNA, you have an I, inosine. And from the previous Wobble pairing table, we know that at third position or at the first position of anticodon, if there is I, this inosine can easily form a pairing with adenine, uracil, as well as cytosine. Therefore, one single tRNA with a single anticodon IgC is capable of binding all the three codons for the amino acid alanine. This is the beauty of Wobble pairing. And this is why we have few number of tRNAs while having multiple codons for a given amino acid. The significance of Wobble hypothesis is that it reduces number of tRNAs required, speeds up protein synthesis, and maintains accuracy with flexibility. Now, there are certain limitations to Wobble hypothesis. Wobble is not unlimited. For example, there are some amino acids such as tryptophan, which is coded by UGG, and methionine, which is coded by AUG. Now, these two amino acids are coded by one particular codon. Therefore, an exact matching tRNA with proper anticodon is there. For methionine as well, only one tRNA is there. Therefore, Wobble doesn't apply to such amino acids. Then, a Wobble at the third position may lead to inaccuracy during the protein synthesis. So, these are some of the limitations as far as Wobble hypothesis is concerned. So, that's all about Wobble hypothesis. It explains how one tRNA recognizes multiple codons, the flexibility at the third base of the codon allows a single tRNA to match to multiple codons of a given amino acid. So, Wobble hypothesis says during codon and anticodon pairing, first two bases of the codon pairs with the anticodon strictly by Watson and Crick pairing rule. Third base can have some Wobble pairing which allows some kind of mismatch to be tolerated at the third base of the codon. And actually this Wobble allows one tRNA to have binding efficiency to multiple codons of a given amino acid. So that's all in this Wobble hypothesis video. We'll see you in the next episode of Biochemistry Insight. Thank you. If you have any questions, you can post it in the comments. I'll try to answer each and every question of yours. See you in the next video. And please do not forget to share, like, and subscribe. Bye bye.